Hi everyone, today we're going to learn about proteins and their structure. So from the study design, what we need to know as part of this component is that amino acids are the monomer of polypeptide chains and result from a hierarchical structure that gives rise to a functional protein. So essentially what we're looking at is how we build proteins and then how we fold them to make sure that they are the right shape to do the job that they have to do. We're also going to look at the role of the rough ER, the Golgi and associated vesicles in the export of proteins from a cell. So we know that proteins have got lots of different roles within an organism. They can have a structural role, they can be enzymes, they can be contractile and involved in movement, they can be part of the immune system as immunoglobulins, hormones, receptors, and involved in transport. So it's important that we understand that proteins don't just have one function. There are many different types which serve many functions in the body. So it's important here to focus in on understanding and remembering a couple of examples, and an enzyme is always a great example of a protein. Proteins are made up of amino acids. So when we talk about this idea of monomers and polymers, amino acids are the monomer for proteins. They're linked together during the process of translation, which we've learnt about already. Proteins are really important for the function of our bodies, and they pretty much create everything in our bodies that we need in order to be able to survive. You need to understand, know, and be able to draw a general amino acid structure. So the way that we draw this, or the way that it's set out, is that we have an amine group with a nitrogen and two hydrogens, a carbon in the middle, which is attached to a hydrogen. Then you'll see down here an R group. This R group is the thing that varies between each amino acid. So it's the thing that's different. The R group changes for each amino acid. Everything else stays the same. We then have a carboxyl group on the other end. So we need to know that we've got an amine group here at the front, our central carbon in the middle attached to a hydrogen and a variable R group, and then a carboxyl group at the other end. And we need to be able to draw that structure. Here's another image showing the different groups in some colours so that it's a bit easier for you to understand. Never fear guys, you don't need to remember all of these, but this is just included here to show you that some of the R groups, the variable parts of an amino acid, they all differ obviously. Some of them are quite small and some of them are quite large. Okay, some of them carry a charge or a polar, while others don't carry a charge and they're more neutral or non-polar. This is important in understanding later on because the charge of each amino acid and where they're positioned in a protein chain impacts the way in which it folds. Connecting amino acids to form these polypeptide chains occurs by a condensation reaction. So when we join together each of these amino acids, it's called a condensation reaction because water is released as a byproduct of the reaction. So we can see here what happens when we join these amino acids together and form these bonds is that these two groups here join together to form a water molecule, and the carbon that's got the double lines or the double bond to the oxygen joins to the amine group on the next amino acid to form what we call a peptide bond that you can see right here. This diagram here shows you how the condensation reaction occurs to form our peptide bond and release some water as a byproduct. So we say that it's condensation when we put the reaction or put the two amino acids together, as you're about to see right now. There we go, water's been released. And the opposite when we break down amino acids is a hydrolysis reaction because we require water in order for that reaction to occur. This diagram here is a really nice one. It shows you where the water is coming from, or how the water is formed in these condensation reactions. And then down here, we can see the formation of that peptide bond. Okay, you need to be able to draw the general structure of an amino acid. You also need to be able to draw what it looks like when that peptide bond is formed and be able to identify the peptide bond. Here's a great question here that I'd like you to complete as part of your Edpuzzle review. So once we've joined all of these amino acids together, we begin to see the formation, or it goes through the process of bonding in different ways to form different structural levels of proteins. Because proteins are very big, we break it down and look at 4 different levels of organization for a protein. The first level is what we call our primary structure. And this is simply the specific linear sequence of the amino acids that have been joined together to make the protein. Our secondary structure is when we start to see some initial folding of the amino acid chain that comes about because of hydrogen bonding. So amino acids in the first instance can fold in 3 different ways that help to stabilize the structure. They can form what's called an alpha helix, which is like a repeating subunit of twirls, a beta pleated sheet, okay, which as it suggests is just a pleated arrangement, or a random coil. So you need to know that in a secondary protein structure, these are the 3 types of folds that can occur. These are some more detailed images of those 3 different types of folding with the beta sheets, the alpha helices, and the random coils. Proteins then have a tertiary structure. So a tertiary structure is referred to as the total irregular folding of the polypeptide chain into a 3D shape. And this is really important because we know that the shape of a protein allows it to be able to function or do its job. So this tertiary folding starts to see amino acids fold over, or the protein chain fold over itself. And this comes about because some amino acids, as we saw before, carry a charge. If you've got 2 positive parts of an amino acid, they're going to fold away from each other. Whereas if you've got a positive area and a negative area, they're going to be attracted together and fold inward on itself. So when we talk about tertiary structure, hydrogen bonding is really important here. The ionic attraction between the R groups, as I indicated previously, is also important. And the hydrophobic R groups, so the ones that don't like water in the protein, also fold inwards so that they're less exposed to the water. And we also see the formation of covalent disulfide crosslinks, which help to stabilize that structure as well. The final level of protein structure is our quaternary structure. This is where we see 2 or more polypeptide chains interact with each other to form a functional protein. So this is where we literally have 2 different amino acid chains joined together to form a functional protein. This is not the case for all proteins. Not all proteins will have a quaternary structure, but many do. We can see here in hemoglobin, we've got 4 different polypeptide chains joined together to form a quaternary structure and therefore a functional protein. Here's another question from B-Card that you can practice as part of your Edpuzzle viewing. Another key term as part of this unit is the term proteome. So you need to be able to tell the examiners and your teachers that a proteome is a study, or the study of a proteome involves the study of all of the proteins in a whole organism and analysing the way in which they act together. This is really important because no protein acts in isolation. All proteins in our body work together in order to bring about a function. The final thing that I want to show you today is just to revisit this idea to sum up the protein secretory pathway. So we've learnt about now the DNA, which is housed inside the nucleus. We've looked at how that DNA is transcribed to RNA. We've looked at how that RNA then travels to ribosomes, where it's translated. At the endoplasmic reticulum, there's some folding which occurs and some finalization, so some groups that are added to a protein. The protein then travels to the Golgi apparatus, where it's packaged, ready for export. Once it's been packaged into little vesicles or vesicles, it then travels out of the cell in order to be delivered to the place it's needed to do its job. And finally, another question for you to practice as part of your Ed puzzle. Thanks everyone.